Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog. Good evening and welcome to Contemporary Science. Tonight we welcome Wong, Creative Director of the Broad Institute of MIT and Harvard. He's also an adjunct assistant professor in art as applied to medicine at the Johns Hopkins University School of Medicine. Wong is also a founder of Clear Science and a board member of the Association of Medical Illustrators and also a board member of Science for the Public. Mr. Wong is one of the bright stars in the field of medical illustration, especially in genetics. His work has appeared in all the major biomedical journals as well as in television programs such as Nova. More important, he's recognized as a leading innovator in making the genetic world accessible to the non-specialist and hard data accessible to both experts and non-specialists. At the Broad Institute, he led the design and development of the very popular DNA trium and data stream. Links to those projects can be found on our website. Wong started out in immunology, but while working on his master's at Johns Hopkins, he got increasingly involved in medical illustration, and ultimately he shifted to medical illustration and proceeded to a wonderful career. He's given lectures all over the United States and the world, and he's here tonight to explain an important field that most of us are really not aware of. We're very honored to welcome Wong.
Thank you. I'd like to show you how I visually represent scientific information for communication, explaining things that we know about science, and also for research, exploring data that we yet know something about. Let me start with this picture here. This simple representation shown simply as a line with two labels on the end. It stands for one of the most celebrated molecules in biology. And does anybody in the audience know what this stands for? This notation stands for DNA. And if you know something about DNA, you know that it exists typically as a double strand that runs in opposite direction from one another. And these two strands are then held together by chemical bases. And there's about 10 of these rungs per turn. And then this whole thing is then twisted into the familiar double helix that we see depicted in the newspaper, leaving a major and a minor groove. And so that's a lot to pack into a very simple representation, but that's really the power of visual conventions that allows communication to be highly precise. But you know, like verbal jargon, convention is really also another learned language. You need to know how to read this. And these two representations, I think they serve the extremes of a continuum, and these ends of this continuum can also represent other things like language, for example, whether we use more metaphorical language to explain science or whether we use more precise, direct language. It can also represent scientific accuracy, whether we have to stick to strict scientific accuracy of scale and resolution and so forth, or can we just sort of highlight the salient points? And there's been a rich tradition in science of using images to communicate and advance science. You know, it's the way for us to show others the sort of the unseen. And this is a painting by David Goodsell, and it's showing a cell at this so-called mesoscale. It's a level of magnification of field of view that's really beyond the reach of modern microscope. Because you're really seeing the forest and the trees all at the same time. And so the way that David paints these pictures is he incorporates lots of biochemical data about the density of these molecules, their shapes, and so forth, to give us this beautiful view of a cross-section of a cell, of a mycoplasm in this case. And you know, it's scientifically accurate and also really beautiful to look at. And he renders these actually in watercolor. This is Albrecht Dürer's rhinoceros from 1515. You know, the interesting story about this is that Dürer actually never saw this animal, and unfortunately it died in a shipwreck when it was being brought to Italy. And so he drew this, he made this woodcut from verbal description and sketches from an unknown artist of an Indian rhino. And it was for, you know, another 60 years until a second species was brought to Europe that people came to know this mythical animal through woodcuts like this one. Ernest Haeckel, German biologist and physician and artist, sort of did the same thing with sea life, showing us this sort of the beautiful symmetry and the variety of sea life in these beautiful paintings. Another German artist, probably less well known to many of you, is Max Bredel. He's known as sort of the father of medical illustration. And in 1894, he came to Johns Hopkins in Baltimore to work with surgeons to illustrate pioneering surgical techniques. And on the right is a carbon dust drawing, a technique that he's well known for. And in 1911, he established the first graduate program in medical illustration at Johns Hopkins. And I'm a graduate of that program. And this is the first assignment that I did in school using this age-old technique of carbon dust, which involves taking carbon pencils and then grinding it down to a fine dust. And then you apply this dust with paintbrushes onto paper. And then the way that you get these highlights, to get the details, is that you use intact pencil that you haven't ground down, and also using the eraser to erase back to the whites of the paper. And after about 40 or 50 hours working on just this one drawing, the way to fix the carbon onto the paper is to use a mouth pipette and blow alcohol across this piece of paper. And it was a very nerve-wracking process. It's very messy. And you know, most of us today prefer to work in Photoshop with multiple undos, right? But I think there's, but despite the convenience of computer, there's many advantages of working by hand still. And this is a drawing of an arm that was done through observation in a cadaveric dissection lab. And I think sketching is an excellent tool for sort of thinking and working through problems that might exist in the real world that you can work through physically. And a sketch like that then can be taken into something like Photoshop and using that tool to emulate traditional media like opaque paint in this case to paint that. So what you're seeing here is an illustration of an arm and how the blood flows around the elbow, because when you flex your elbow and you leave it straight, the blood still has to get through, and there's many routes for it to get through, and this illustration illustrates that. One of the challenges in medical illustration is drawing what's sort of difficult to see, and the recruiting brochure that is commonly circulated to advertise medical illustration says something like, draw what can't be seen and tell thousands about it without saying a word. And one of the subspecialty of medical illustration is surgical illustration. And not only is it difficult to see, but the challenge is to depict what the surgeon sees when you're not standing where the surgeon is standing. So you might be standing at the head of the table, but you have to imagine what the surgeon is seeing when he's standing at the side of the table. And it was in surgical illustration that I learned about a concept called the surgical moment. And it's a concept that I like to describe to you. So a surgical moment is a pivotal moment in surgery, and where relevant anatomies have been identified and protected or prepared for some subsequent manipulation. And what you're seeing here is a step in the Whipple surgery. It's for the treatment of pancreatic cancer. And in this procedure, portion of the pancreas, the bile duct, the gallbladder, and duodenum is removed. And then the remaining structures are then sutured back into the intestine to direct the secretion of the digestive enzymes into the gut. And just in this one illustration, you see that the duodenum has been closed off, the pancreas has been dissected, the branches and the arteries and the veins have been tied off, and the neck of the bile duct has been exposed. And medical illustrations are trained to capture these moments. It's because it's with this type of illustration that many surgeons still learn and teach surgeries. If you're squeamish and that looks bad to you because you just had dinner, you might want to check your email for this next slide. Medical illustration is typically compared to imaging technology like video or photography. And, but hand drawing illustration still remains the best way to really communicate surgical procedures, because in a photograph, you see what you get, and no more, no less. And you know, good photography depends on good lighting, and in the OR, the surgeon controls the lighting, not the photographer. But with illustration, we can selectively leave out things and only focus on the things that matter. You know, when you're learning to do surgery, the blood doesn't really matter and fat doesn't matter. It's like the really important anatomical structure that matters. And so when you do an illustration, you can just focus on those things. And I think one of the real benefit of illustration is that you can show sort of a moment in time. Whereas photography, you get to sort of like this instant in time. Here, there's many instances together. This instant probably never existed. It's sort of a moment that capture seconds at a time. And, but it's this ability of the artist to decide what to show and how to show it, I think is going to continue to be important as we move forward in the sciences. My work at the Broad today is very much unlike that of a typical medical illustrator in content, but my approach to designing figures, scientific graphics, and data visualization is very similar. It's a process of sorting through information, deciding what's relevant and what's the message for that communication, and then using the same sort of graphical techniques of color, composition, and typography to get that message across. And when we do this, audience is really paramount. So if we were to design a teapot for a masochist, you know, we would have to take special considerations in mind. And but what the research audience really cares for is how experiments are done and the reproducibility of that results. And so these types of graphs and diagrams that we see in peer-reviewed journal are really the mainstay of science communication. Figures are really the currency of scientific communication, and there's one particular type of figure, these sort of overview figures, that are often left out because space is short and people want to just cram in as much data as possible. But these overview figures, I think, are really important because they provide a framework for understanding the experimental design and the reported findings that are shown in the paper. And so I pay particular attention in trying to get these things well designed, very compact, and have them be included in the publications. And you know, at the Broad, I think we really care about the science that we do, and I also care about how we communicate that science. And many of the papers there include, we make space for this overview figure to explain the science and make sure that people can understand it. This is for a different audience, and the audience for the New York Times, they care about different things. They care about the big concepts in science. And I brought this here tonight to show you the importance of scientific accuracy. When this graphic came out, I got an email right away from an old teacher from Johns Hopkins, and he said, saw your diagrams in the Science Times, very nice, but oops, a bit wrong. DNA wraps 1.67 times around the nucleosome, and your diagram shows 2. And you know, with this mistake, though it was immediately noticed, it didn't really compromise the understanding of the figure, really, because it's really about these marks that are on the protein that organizes DNA. But nevertheless, this type of sort of a mistake undermines the credibility of the whole piece when a scientist looks at it. But the funny thing is about this piece is that he didn't notice that the DNA is wrapped around the histone the wrong way as well. But this is what happens when you work over electronic media. Pencil sketches gets translated in different ways, and wrapping something around something else 2 times is much more symmetrical, and maybe it doesn't matter if you wrap it around this way or the other way. But now that we know what an octopus looks like, we would never depict it with nothing but 8 legs. So I think it's important to depict things with accuracy. For a sort of different venue, the Broad website carries scientific stories that are done at the Broad and also elsewhere. And people who come to the website are looking for sort of current research or other news organizations looking for research ideas to carry. And so the images that go along with these stories are less didactic and are more impressionistic. This one here is about the epigenetic landscape in stem cells and how the DNA, the environment around DNA really affects how the genes function and not the genes themselves. This graphic here describes a very cool discovery a year ago or so about how DNA folds in a cell. So a cell needs to fold a really long piece of DNA, about this long, into a tiny space inside its nucleus, and it has to do this so that it can be unraveled at most notice without any knots in it. And it turns out the way that it does this is it uses something of fractal geometry. And it does a space-store filling curve that's described by mathematicians like Hilbert and Piano. And if you remember the old Windows screensaver where the pipes are running and they turned, I don't know if you remember that, but that's exactly a Piano curve, a Hilbert curve, and the cell's using that compaction to fold the DNA within the cell, and that's how it's able to get a really long piece of DNA in a very short space, and then also have it be able to fold apart. And you might have wondered, why the crumpled piece of paper, and the crumpled piece of paper is actually the simplest example of a fractal, because large pieces of paper and small pieces of paper will give you about the same proportions of large and small facets, and so I like to design images with this type of propositional density, where there's sort of layers within layers of meaning. And I think only one mathematician got that inference to the crumpled piece of paper, but I don't know. But I still like to do it. A few years ago, I was asked to design a cover for Nature to commemorate the completion of the sequence of the dog genome. And I find these projects really challenging because, like figures, everything is actually there. You just sort of pour it on the table and sort of sort it out, sort of create a hierarchy, what's important, what's less important, prioritize the information. And also the purpose of the figure also guides you. In this case, if it's a comparison, you want to just show everything that's alike and only highlight what's different, because that's what you care about. And if it's a process, then you show, you leave visual anchors from one step to the next. So you can sort of see what's changed as the progression goes. And this is something that comics do very well. And this is actually illustrates, this is another overview figure published in Science that shows you the high C technique of how they figured how DNA folds in a cell. But for the dog genome, though, there's really not a lot of starting material. There's a dog that you have to include, and then sort of some representation of DNA, and that's all you got to go with. And the dog genome is actually scientifically very important because many dog breeds many dogs get the same disease that humans do, and certain breeds are prone to certain types of diseases, and breeders have kept meticulous records of their breeds. And therefore, it makes the dog genome really a treasure trove for looking for genetic causes of human diseases. During this time of this cover, we were celebrating the 50th anniversary of the discovery of the structure of DNA. And this picture of James Watson and Francis Crick around the model really was the sort of quintessential image that's about DNA, that's discovery of DNA, and also about the sort of the double helix nature of this. It's just that there was one problem, which is there's no dog in this picture. And so with a lot of cajoling of the editors at Nature and permission from Watson himself, we added that dog into the picture. And with the white shirt of Watson sort of mirrored the white chest of the boxer whose DNA was sequenced. And so the theme became sort of mirrors within mirrors, dogs looking at looking at DNA. And so here's the sort of final cover for that. And we were talking before the show here that this image didn't actually run with a byline that says this image was photoshopped, and so I think if you look closely, you might not have. If you don't look closely, you might not notice the dog. If you do look closely, you notice the dog there. But probably we should have a caption so that to say that it wasn't Watson Crick plus dog that discovered DNA. So the DNA trium, another endeavor for science for the public. Institutions like the Broad where I work, I think they have a responsibility to tell the community about the science that's taking place there. And research of the Broads involved deciphering all the information that's encoded in the human genome and understanding that human genetic variation and its role in disease. And communicating this science effectively is a challenge, really, because there's really few channels to do this, and it requires a lot of dedicated resources. We've tried to do our part by creating something called the DNA trium, which is through a series of unique exhibits we want to enable people to explore the human genome. And it's located in the Broad's main building. It's on the edge of the campus of MIT in Kendall Square. And the audience really are visitors to the Broad and then high school students who come for different programs there. And then just foot traffic on Main Street because the Kendall Tea Stop is there. And there's a lot of lunch places where the lunch crowds sort of pass by. But translating current research for this audience, we have to shift our approach on that continuum that I described at the beginning of using new representation and relying less on conventions, and then using more metaphorical language and less sort of direct and archaic language. And one of those exhibits that was alluded to at the top of the program is the DNA trium. And the Broad makes much of the data it generates publicly available. And the idea was, could we just sort of take that same data and metaphorically stream it onto the street? And so that's what we've done here in these large panels that you can see on the inside of the lobby as well as outside the street. And I think what's really exciting about this project is it gives the public a chance to see raw data. Oftentimes we just hear reports in the newspaper about some sound bites about a scientific discovery, but if you're trying to trace that back to all of the data that was generated to get there, you can't do that. And so here we present the raw data, and not so much about the discovery that comes from it necessarily. What I'm showing you here is one of those panels. This is on DNA sequencing. And the way that we sequence DNA is we labeled the 4 genetic letters of DNA, A, T, C, G, with a different color. And as that DNA molecule passes by the detector, we read that sequence of color. And it was in this way that the human genome was sequenced. All 3 billion letters of it was sequenced. And so what you're seeing here is the work of one machine. And it's able to do about 100 samples, 98 samples at once. And what you're seeing in the upper right there is the detailed trace of that color sequence call. So it's saying green, 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 AAAA, GT, so forth, and it just keeps going like that. But and the reason that I wanted to show this is if you compare the peaks now, they're very dull and they run sort of together. It might be hard to see from your seat, but when you see a band of greens like that, is that 5 A's or 3 A's? And the computer has to continue calling that. And it's because as the enzymes involved in doing DNA sequencing gets tired, its fidelity is really low. And so this is actually telling something about, it's not just data, but that's this idea of data quality. This is poor data. And if you compare that to the beginning, you can see that the peaks are very sharp, very high, and this is high quality data. I don't make a long discussion about that, but I show the raw data, and there's this, he puts out there that science is not necessarily very sterile and clean. There's this sort of dirtiness to science. I'll show you a couple more because I think these are very interesting because they give you a different sort of feel about science. This one is on genetic analysis. Remember I told you that the human genome is 3 billion letters long? And we've heard these stats sometimes that any two individuals is 99.5% alike. And that means that there's a spelling difference every 1,000 bases or so. And this collection of spelling differences is what makes up our genotype. And the way that, and these variations is what may account for our different susceptibility to disease. And the way that we read out the genotype is on something using a gene chip. And what you're seeing here is the probe that's being read, the variation that's being read, and then it's mapped to the corresponding position on our in our genome. So our genome is made up of 23 pairs of chromosomes, 23 from mom and 23 from dad. And so what you're seeing here is over time, over about a course of an hour, 1 million of these sites are cataloged, and you get this sort of complex fingerprint emerge. This is data from a study about HIV, and there's a group of individuals that's been exposed to HIV but don't develop AIDS. And so they're very important to the medical community. And so by looking at their underpinning of their genetics and comparing it to people who are exposed to HIV but develop AIDS, it gives you sort of an insight onto the mechanism and the genetics that's involved in that disease. And the third area that the Broad's highly involved is in chemical screening. And chemical screening, it's about sorting through thousands of compounds looking for a hit, something that may stop a cancer cell from growing but leave healthy cells alone. And what you're seeing here is the compound being tested and then its effect on the cells. And I think what I really like about this one particularly is it's really inviting passersby on the street to participate in this process of discovery, because you're seeing exactly what the scientists are seeing upstairs, and we tell you sort of what to look for, and you're looking at the same thing. Of course, there's a lot of tools you can bring to this, like automation and computers and so forth that's doing the looking. I mean, there is some individuals doing the looking, but most of the looking is being done by the computer. A nearby exhibit shows you the technology that's used to generate that data. In this case, this is a DNA chip reader, not too unlike our desktop computer. It's not very exciting. But I think the important thing about this exhibit is it also lets us have a chance at portraying scientists in a different sort of way. This is, if you Google scientists if you Google scientists and use images, this is the top hit as of last week. And I think when programs portray scientists, sometimes they portray them as these sort of extraordinary character that is really hard to relate to, like Einstein or some mad scientist. Or they go way out of their way and say that, a scientist could actually be your next door neighbor. They also go to the movies and go to coffee shop. But then they seem sort of bland. And so we didn't want to focus anything about the person, but just about the individual and their sort of passion for research. And I think that's I think people can relate to this, that, I don't care if they go to the movies. They're really passionate about their work, and that's what we do here. And I think that also allows us to show people the faces of the people who show the audience faces of the people who work in the building, because oftentimes science is practiced deep within campuses in these sort of windowless buildings, and you have no idea what goes on. And so this is bringing a bit of the lab and the people to the window. But really the centerpiece of the DNA trium is something we call the CRX. And it's a media wall that's in the shape of a spiral. And it's an interactive display wall that shows news from the international research community. And it's comprised of 80 42-inch screens that are connected together as one sort of graphical space. And really the physicality of this is really part of its attraction, because you put these sort of same stories on a website and it doesn't have the same appeal. But if you stand in front of this thing with a controller, and you can sort of feel the heat and the immensity of this thing that's 10 stories or 10 feet high. It has a sort of a different impact, and so it's been fantastic to be able to bring this type of news story to present it in the community in this way, because you can control it from the street as well, and so we know that people are reading it at 3 in the morning. I mean, they could be MIT kids coming home from parties, we don't know, but at least it's being used in the middle of the night. So somebody's reading about dog genomes and so forth at that hour. So most of the exhibits at the DNA Atrium can be controlled from the outside, except for this one, which is a touch table, and you do have to come in to use this one. But I like this interface because it's not just multi-touch that we're used to, but it's also multi-user. So many individuals can work together on a project. And what I'm showing you here is a timeline of medicine, but we've also are trying to develop simulations so that you can sort of recapitulate the process of science. How do you go from taking a large database of numbers and then trying to find the genetic root of disease? Like how do you go about doing that? So these simulations that we'll build will enable people to do it that way. So many of the graphical techniques that are used for communication can also be applied for depicting data for analysis and trying to get something out of the data. And in this review that I wrote in 2010, many areas of biology data generation is being replaced by data analysis as the rate limiting step. So it's very easy to generate data. It's very difficult to try to make sense of that data. And you know, patterns are the essence of really data exploration. I think visual representation has an important role to play in making sense of that data. And let me give you an example here of why we might want to visualize quantitative data. So this is Anscombe's quartet. These are 4 sets of numbers here that are statistically identical. And if you look at this table of numbers, nothing really stands out. And if you calculate their stats, they have the same mean and the variance on both the x and the y, the same correlation, the same linear regression, and 16 other common metrics that we use all the time. But when you go to plot this, data, it just takes a glance to see that they are wildly different. Like this is, you know, and this is really the power of visual representation, because it affords us a penetrating look at the structure of data. But when we represent data for analysis, it requires a slightly different approach than those used for presentation, because in a presentation the message is clear, and the purpose is really to get that point across, and we can do that in many ways. We can emphasize certain aspects of that thing. We can use color, for example. We can use size differences, or we can employ sort of simple movement to attract the viewer's attention. But when we encode data for exploration, it's unclear up front which aspect is most important. So the more appropriate approach is really to encode the data object with some equal visibility across, but not to have any one aspect overpower another, especially inadvertently. And one of the things that people love is color, because it makes things very colorful. And this is a brand new building that was built. I spent the summer in Denmark, and this is a new building where I was situated. And in the course of my stay there, they erected these 4 columns that had no function at all except to have this pattern painted on it. And so we really love color when we encode data. It's really the thing that people typically reach for first, the graphical variable that people reach for first. This diagram is taken from the website of the National Human Genome Research Institute. And it shows us the 23 pairs of chromosomes and all the genetic variation sites that's been cataloged. And this figure here is using 237 distinct colors to depict the 237 disease-related variants. It comes with a key, so if we try to look for this color here collectively as a group, this sort of purple color, and we turn the page and we look at the key, we could try to find it, and you know, it could be that one, or it could be this guy down here, or it could be any one of these. The reason I want to show this is to make a point that when we encode data graphically for communication, it's really a two-way street. There's the encoding process, which is the way that we choose to represent the data. And then there's the reading part, the decoding part from the audience of how they interpret what we encode. And I think if there's any part of these two parts of the process is compromised, the whole communication really suffers. So there's an expressive bit where the author puts something down, and then there's the decoding bit. The effectiveness bit is in the hand of the reader, and can they interpret what we intended that information to convey? And in this case, I think it's really tough to do that. But color is very attractive, and so it's the thing that we reach for first. The problem with color is that it's one of the most relative medium in art, and our perception of color really depends on the neighboring area around that color. So the lightness of these two inner squares are the same. And even if I tell you that they're the same, it's really hard to overcome what our eye sees. And until I really sort of take and put them together, that you see that they are in fact the same shade of gray. And simultaneous contrast is what causes this visual phenomenon, and also affects color. And again, the inner squares here are the same shade of green, but they appear different. And you know, if we bring them together, you can see that in fact they are the same. And in this sort of very way of making the same color appear different, you can make sort of different colors really appear the same in this case. So color is very challenging to represent data, but one of the most popular form of data representation in genomics are these things called heat maps. And heat maps rely on putting a bunch of colors next to each other. And what a heat map is imagine a Excel table with a bunch of numbers. And then you say 3s are red, 4 is a deeper shade of red, and 5 is a really deep shade of red. And then -1 is a blue, -2 is a deeper blue, and so forth. And then you encode the whole table of values based on color. And they're widely popular. If you flip through, so this is a picture of the top 4 science journals, Cell, Science, Nature. And you can't turn the page without seeing a heat map in it. And they're ubiquitous. And they're fantastic because it allows you to crunch a lot of data into a very small space. If you look at this image, you know, that's probably a 10,000 by 10,000 matrix in an Excel table or something like that. But it's hard for us to make sense of it. This is a simpler one that was published in Nature as well. It's a heat map here. And the two asterisks indicated two cells here in this color table, and they're in fact the same, but they of course appear different, and you know the drill now, so if we bring them together, they're actually the same. So this really popular form of communicating science data actually biases scientific data. It takes the same underlying discrete value and it makes it look different to us. So color can be a big problem when it's used to represent quantitative data. Researchers also occasionally use the rainbow color scheme because it makes these sort of fantastic sort of pictures. But even, you know, if we remember the mnemonic Roy G. Biv, it's still really hard to answer, like really quickly answer questions like, you know, is yellow bigger than green, or is blue bigger than red? Or, you know, it's hard to see that. But I think there's another problem that underlies this, is that the transition in color in a rainbow color scale quickly breaks its correspondence with the numerical scale. So let me show you what I mean here. Like if you look at the rainbow color scale and you look at this portion of it, an increment of 1 is a change from this light green to green, essentially. And if you look at the same increment on another part of that scale, it goes from yellow to red. And in this way, we also are biasing the eye, and you could shift this color scale so that these patches of things are really jumps out. But we're cheating here a little bit because this is an even scale, but qualitatively we've just shifted it to something else. So we have to take in account of what the eye sees, you know, and color is also prone to color blindness. And you know, there's some stat that says if a manuscript in Science goes to a journal, and it happens to go to 3 male reviewers, which is not that unlikely, the chances of one of them being colorblind is 23%. So it's almost a quarter of the time your manuscript is going to be seen by a reviewer that can't see what you're communicating because you've used colors that are not friendly to the colorblind. But color is only one way to depict quantitative data, and there are many ways that we can do this. And in this figure here, I'm showing you that many different ways of depicting the same 5 values. We can do it with color hues, like the rainbow color scale. We can also do it with color intensity, light to dark. We can also use volume and area, or angle of a line, or length, or position on a common axis, or position on axes that are not aligned. And in 1985, in a science paper, Cleveland McGill actually tested people's ability to extract information encoded in these different ways. And what they found was that color hue fared the poorest, not surprisingly, because there's no natural ordering to color. And at the top of this list, it's things like position on a common axis, like your typical XY chart. You can pick those things out very well. And things like pie charts that are very popular as well depend on judging angles between things and area, perhaps. And those are also sort of lower on the scale here. Any bar chart can be replaced, any pie chart can be replaced with a bar chart, and you can see nuances in the data. So a table like this is very useful when we occasionally have to depict come up with new representation for data. We would want to rely on things that are much more accurate and efficient at the top of this list. And I'd like to give you one sort of a real-world example of how these sort of ideas of relative colors and different ways of encoding data can be put into use. So some researchers at the Broad study functional genomics, which means that they want to study that in a given molecular network, points in that network that's been preserved over evolution, and points in that network that has been not. An example of such a network is glycolysis, how we metabolize sugar in our body. And on the right there is a depiction of that pathway of how sugar gets converted to energy. And when they started, they took that same, it's drawn differently, but all the connections are the same. They took glycolysis, and what they did was, let me explain the representation here, which is that the metabolites, sort of like the sugar and so forth, are represented by the boxes, and the arrows represent the enzymes that take that metabolites, convert it to another form, and so forth. And so what they did was, across all of the species that they studied, in this case was the yeast was the model organism, they found points in that network where all the species behave the same, and points in that network that they behave differently. And what they did was they encoded that with color. So red meaning where everything's preserved, and blue meaning that they were not preserved. But if you look at this and you try to order it, it's difficult to do. What's most preserved and what's least preserved, and you put this in order. Maybe you can pick out the two ends, the red and the blue, but then everything in between is hard to see. I think another way to depict this is you can actually sort of linearize the pathway. And still present the general connectivity of the pathway, but this way you've just created a common axis. Remember, a common axis is very useful. And instead of using color to depict similarities, you can use length, which is also good. So the longer the bars, the more preserved. And in this way, you can easily ranked order these bars by length. And this economical representation also allows us to encode data about the enzymes, the arrows that connect the metabolites from one to the next. In this case, we used dots there, which disappeared very quickly. And having both together. So if we represented the enzymes in the old depiction, that means adding colors to all those arrows. So we add a lot more colors. In this way, we have them together, but you can sort of ignore the lines. Focus on the dots, sort of ignore the line, but having them together improves the utility of the data that's being presented. So bars for metabolites and circles for enzymes, and you create this sort of visual layer that you can selectively look at one or the other. And but in this study, it wasn't just one point in time, because that would be not enough, because you're trying to look for how things change. But so they've collected data over time through the life cycle of a yeast. So this sequence of color represents the life cycle of the yeast, how things change. And when you look at a sequence of color, it's hard to tell the trend. But if you actually plot that using one of these other encodings, you can see that much easier. So here I'm using a filled line curve, and filling that area under the curves to exaggerate the shape of it. And if you do this across color depictions, you can see that it's much harder to see. The color sequence is complex. You can't really tell what's happening, whereas there's a dip and whereas there's a mountaintop, trough and peaks. But you can easily see that in the profile curve. And you know, if you look at a few things, it's fine. But if you look at hundreds of things, it gets more difficult. And I think this depiction using these curves allows you to see certain types of patterns where you have inversions, or you have a peak that's shifted from one point to the next. Those types of things are much easier to see, and it allows the researchers here to, in this case, pick out the time in evolution where two genes diverge, where you know, certain species took a certain path of using that gene, and another species took that gene and did something else with it. And so this is a tool we created called Pathlines, and we presented this in Bordeaux last year at a conference that's about visualization. So one last vignette about art and science, and making visual representation of scientific material is one type of art. In this project, we took the phylogenetic tree and turned it into a phylogenetic mobile. A phylogenetic tree tells us how animals are related through evolution. And so it's, you know, if you turn the phylogenetic tree this way, you can sort of easily imagine it as a mobile. And in doing so, we sort of solve one of these data presentation challenges as well, which is that things that are sitting next to each other are typically read as being most related. But in fact, if you trace them up the tree, they could branch off into different areas. In a 3D structure, in a mobile, things that are sitting next to each other but belong to different branches would just simply swing further apart. So this was completely serendipitous that we said, we'll just build a mobile because it'll be really cool. And then, but I think we solved one of these problems with the 2D representations of phylogenetic tree. And so what we built was a phylogenetic tree of the 33 mammals whose genome has been sequenced. And the edge length in the tree, it represents evolutionary time. How far is the extant species, the species that exists today, how far is it genetically from its common ancestor? And all of those lengths have been preserved in this scale. And so what you see is really sort of interesting things that comes out, like that the cow is actually more related to the dolphin than it is to the horse. And I think when you visually depict these things, interesting facts like this comes out. And if you look at this tree, if you look at this mobile, you can sort of see that there's 4 major branches of mammalian evolution, and you know, we're here on one of those branches, and so where the other animals are. So from that model scale, we built the full-scale piece that's hanging in the lobby of the Broad. Now we're up to 36 mammals have been sequenced, so this thing's outdated. Only shows 33. Can't keep up with science. But I think another, you know, that's one form of art, which is just taking scientific information and depicting it somehow. But that's sort of another form of art is also supported at the Broad through our Artist in Residence program. And the goal here is to really foster the discourse between artists and scientists, and for to enable the other to see how each other work and solves problems. And you know, the process of art, I think, is not so dissimilar to the process of science, and scientific discovery doesn't happen at a moment's revelation where somebody hits their head and has an aha moment. It goes in spurts, and then progress is followed by setbacks, by failed experiments. And art doesn't happen in a flash of creativity either. If you talk to artists, they talk about these sort of sparks of creativity, and then it's followed by the loss of imagination, and they're stuck for months. But I think what's really nice to bring these sort of two worlds together is that they can sort of share their worldviews. An artist's worldview comes through his or her art, but it's very different perhaps than the scientist, and the scientists may see the world differently. At the Broad, if you talk to genomicists, they say, DNA, that's the center of all things. After all, it carries the genetic information. But if you go down 3 floors and talk to a chemist, they would say, oh, DNA, that's just the brick and mortar of the building. It doesn't mean anything. It's really the small molecule that's firing neurons that allows you to think and have conversation and memory. That's the real stuff. And I think sort of in this way, artists can really help scientists do better. Artists can help scientists do better science, and scientists can help artists make better art. So I've shown you a series of vignettes tonight, from medical illustration to scientific graphics and visualization and art. And the thing that ties all of these things together is representation, specifically visual representation. And I think that there's this sort of powerful connection between thinking, seeing, and understanding. And so that by considering the principle from art and design, and we can improve information transmission, that remembering that expressiveness only depends on our choices of graphical means, but effectiveness also depends on the perceiver. And sort of by considering the need of the audience, whether our colleagues or professionals in other fields, we can extend the reach of our communication. And then by considering this idea of the surgical moment, we can create images that have sort of a landmark for sense making. And finally, from drawing from many disciplines, like from art and so forth, we can enhance the richness, complexity, dimensionality, and clarity of content. So with that, thank you so much for your attention. Sure. Yeah. We have some time here for some questions. Okay. Yes.
How did you get the mobile to balance? Sorry. How did you get the mobile to balance? That was amazing.
There was a trick, which is that in the phylogenetic representation, the horizontal beam doesn't carry any information. So you can put the level, the higher level, at any point there. And in the large mobile, we had to cheat a little bit more and use sort of lead weights in some of the animals, because you have elephants and you have mouse. So, yeah, in the front.
What is the what's the newest animal in the DNA? The mobile.
The newest one to be sequenced, or the most?
The newest one to evolve.
That's a trick question. I don't know the answer to that. So in some ways, it's the question is what's most evolved, right? And I think a human-centric researcher would say humans maybe, but I think we have to look at the DNA. So I think there's some comparative analysis there, and I think you have to consider what it means to be most evolved. And there are some deers with only 4 chromosomes. Are they less evolved than a fruit fly who has 16 chromosomes? I don't know. So I think it's a tough question, but I don't know if I have an answer. This question.
You were talking about putting material into scientific journals and having to work to get all that information in there. Do you foresee in the near future that scientific journals will be online in a way that all this information would be available that the researcher wants to communicate?
There's a mechanism now to put in a lot more information that's what's published in the print copy. There is a companion online that allows you to submit as much as you want supplemental material. But that supplemental material is hard to make sense of unless you're a real expert, that you're willing to download that terabyte of data and look at it yourself. And so I think the journals are working hard to come up with other ways to present scientific information more effectively. I know for a fact that we've been working with Nature on the heat maps and the ability for the author to choose any color scale they want, which they emphasize different things, and for Nature to enforce a certain type of color scale, you send them the data. They always draw the heat maps in the same way. So that way you can compare one presentation to the next. And so I think the journals are working hard at that. You know, still you can't see, even online, you can't see structures in 3D yet. And you know, there's lots of visualization challenges. And it's an interesting question too, because in years past, the figures really had a central place in the scientific journal. It sort of carried all the weight of the scientific finding, because you can look at it and say, I don't really believe what you're saying because that cell looks really fuzzy to me, and I don't, you know, that green-red dot is not where I expect it to be. Or you look at a gel and you say, oh, that band is slightly higher than where you claim it to be. It's pretty fuzzy too, or something. But now you have a half-page figure, and it's one genomic locus out of 7,000 that they found. But this one is supposed to be representative. It's the figure is half a page, but you see one example out of 7,000. And no longer is the figure has this sort of central place in carrying the discussion. So things really shifted to computation, and so in these papers are reported significant values and these types of things that you sort of have to take it at face value in some ways.
You talked about a wide range of representations, from something that's fairly realistic, like a surgical illustration, all the way to more abstract notations representing, you know, various genomes and so forth. How do you feel in terms of the creative process that goes into one or the other of those types of illustrations? Are they the same skill sets and the same kinds of activities, or do you find them completely different?
I think both. In some cases, it's the same. It's like, I think if you're trying to represent a concept that's challenging, I think working through that same concepts can be very satisfying. It's working through a surgical illustration piece or a table of numbers. On the other side, so I think it can be satisfying if you're just thinking about the problem that you're solving visually. But if you're depicting a heart looks a certain way. Number 4 doesn't. You can depict 4 any way you want, as a slice of a pie or a height of a bar. And so I think that in some ways you could say quantitative data needs you to be more creative, because it can be represented in these ways, because it doesn't have a physical form. I hope that answers the questions. Yes. I think it's a little bit. Yeah.
I'm wondering what your role is in deciding how to visualize certain data. So a scientist generates this data and has a particular idea that they want to explain. But you said, as you showed, there are many different ways to show that, and there are ways that can bias the audience's perception. So do you find yourself taking a role in shaping the way data is presented for scientists?
Yes, I think the driving force behind representing scientific information is sort of the difference between art and design. I think I would say art appeals to the emotion and the aesthetics, whereas design really appeals to functionality and practicality. That's my distinction for it. And I think scientific graphics, in many ways, is a design process, not an art process. I mean, we're serving a purpose. We're trying to communicate a piece of information. And in that way, the scientific question drives the design of the graphic. And the graphic is to serve that sort of task. You know, what is it that the researcher needs to see? Are they trying to see outliers? Are they trying to see, are they trying to compare things? Are they trying to see correlation or trends? and you try to come up with the graphical representations that potentially highlight those things more effectively than another representation. And it's really to serve the scientific need first and foremost. And that's what I focus on. So there's another question back there.
When you're going through that exercise of turning some set of data or a principle into a visual representation, do you have a standard set of questions or principles that you always kind of run through, either in a particular order or just want to get to all of them in order to kind of guide you in figuring out what the best visual representation is?
I think there's a couple of things, I think there's this sort of general data analysis. You're looking for outliers, you're looking for correlation, you're looking for trends, and we're always looking for those things. And there's the so you know, I think if you can highlight any of those things, it's helpful. You also take the scale of the data into consideration. If you have 100 points, there's many things that you can do with 100 points. But if you have 1,000,000 points, then you have to take a different tact. And that tact may require some more computation and using dimensional reduction techniques. You know, like for example, taking averages or medians and so forth, just to get those numbers down, and so that you can have an overview, and then you can dig back in deep and try to see all of the data points. So I think there's the scale issue that also drives design, and then sort of the basic needs to see patterns, outliers, correlation, and so forth. And there's a handful of those things. And then that table I showed, I try to stay away from color as much as possible, but you know, and to use it judiciously. I mean, it's great for categorical data, whether it's apple, oranges, you can color code those things. It's tougher for quantitative data.
Hi. If journals go digital, it seems like the road of illustration completely changes because there's so much choice you have there now. You can have animation, or the user can choose the color.
Mm-hmm.
Is there any move to have journals go digital?
Journals are digital.
Well, but
it's just that they take the printed page and they convert it to a PDF.
Well, that's not digital.
So it's not digital.
I mean,
You're right. And I think the journals, surprisingly, it's sort of slow going to get to this point. And I think that there's some effort being made. So I write a monthly column about taking graphic design ideas and applying it to scientific communication for nature. So I know nature the best. And so I know that there's an effort underway to design a sort of a website that's just dedicated to visualization. And I think some of those things then will hopefully make it into the digital form of the journal. But currently, you're right, it's just the PDF version of the printed page, and there's really not more than that. And of course, journals, really, it's the currency of science, and that's what drives science. And whether it's the right model, I don't know going forward. Yeah. Okay. Well, thank you so much. Thank you for your attention.